What do mayonnaise, vinaigrette, and the Clark Lab have in common? They all rely on the process of emulsification. Emulsification is when you bring together water and oil into one coherent mixture. Oil and water are immiscible fluids, which means they don't like to combine with each other. But if you throw oil and water into a container and add enough energy to the molecules, you can encourage oil and water molecules to get close enough together to form emulsions. Emulsions are like tiny microscale droplets of one solution within another immiscible solution. In the case of mayonnaise, mayo is made up of minuscule oil droplets suspended inside watery liquids, like lemon juice or vinegar and mustard, which we have over here. This structure gives mayo its characteristic thickness and creaminess, which we all love about mayo. Usually, emulsification requires an emulsifying agent. When we make mayonnaise, egg yolk acts as an emulsifier. The lecithin molecules in egg yolks have a hydrophilic head and a hydrophobic tail, and they act as a cushion between oil droplets and water. Lecithin molecules actually surround the oil droplets that are immersed in the water and orient themselves in such a way to prevent the oil droplets from coming into direct contact with the water molecules, which would cause the droplets to break apart and break the emulsion. In the kitchen, we have a few different ways to inject energy into the system to start the emulsification process and make a mayonnaise. One way is to use a whisk, another way is to use an immersion blender. It turns out that you can use emulsions as tiny reaction vessels for biological or chemical reactions. If we can trap cells inside droplets, we can do biology experiments on millions of cells all at the same time, instead of having to manually perform experiments on each cell in a well or a test tube. But while whisking or blending might produce very tasty emulsions in the kitchen, these methods of emulsification don't quite cut it in the lab. In the Clark Lab at UC Berkeley, we use a technique called droplet microfluidics to generate and manipulate emulsions. Droplet microfluidics lets us create oil and water emulsions that are all constant in size and precisely tunable. We can trap cells inside emulsions to carry out biological experiments or chemical reactions. We can even string together droplet microfluidic functions to perform multiple operations on the same cell. Microfluidics can be used to generate emulsions, merge two droplets together, inject liquids inside water and oil droplets, or even separate droplets based on specific characteristics into different tubes.
HIV-positive cells are rare, making them hard to isolate and study. They total approximately 1 in 1,000 to 10,000 CD4 T cells in blood. A small subset of these cells is capable of producing infectious virus after treatment interruption. These cells represent the major barrier to an HIV cure. We created a microfluidic method called FIND-seq, FIND for finding HIV-positive cells, SEQ for sequencing their transcriptomes. We start with tissue, and we dissociate it into individual cells. These cells are combined with oil on a microfluidic device to produce water in oil droplets. Microfluidics allows for the collection of millions of individual cells. Inside each droplet, we perform a digital PCR that produces a fluorescent signal only if HIV proviral DNA is present. Droplets with HIV-positive cells become fluorescent. Those with HIV-negative cells do not. We again use microfluidics to sort the positive droplets, to collect them into tubes, and to analyze their transcriptomes. By comparing HIV-positive cell transcriptomes to HIV-negative cell transcriptomes, we hope to understand what makes infected cells unique. With this information, we hope to find vulnerabilities in infected cells and understand why they persist in vivo for decades.
If you want to read more about FIND-seq or other ways we use emulsions in the Clark Lab, feel free to visit our website at clarklab.berkeley.edu or read any of our published papers about the method.